Hi, good afternoon. As Maya just said, my name is Antonio Moore, and I interned with Pacific Northwest National Labs this summer. And I worked on part of the HuBMAP lung program, and I'm excited to share. So the HuBMAP lung program is a multi-center collaboration with the goal of generating high-resolution, multi-omic, 3D biomolecular maps of non-disease lungs. For this project, I worked with the team at PNNL that focuses on spatial metabolomics of the human lungs, and we mostly focused on identifying lung lipid species on specific tissue functional units, such as like airways and alveoli. For this, we used matrix-assisted laser deabsorption ionization mass spec imaging, or MALDI MSI. And so how MALDI works is that we first cut tissue, and we coat that tissue in a organic acid or base dissolved in an organic solvent, and this is called a matrix. The matrix extracts the lipids and biomolecules and co-crystallizes them and incorporates them into crystals. The crystals are then hit with a laser, and where most of the beam energy is absorbed by the matrix crystals, and then it releases the lipids, and the beam also ionizes the lipids. The ions are then collected by the mass analyzer and given a mass to charge ratio. But most of the ions, the ions are most commonly singly charged, meaning that when we do the mass to charge ratio, it's the mass over 1, which basically just gives us a mass. And with that, we're able to get their chemical formulas. So the process of doing this is we first start with cryosectioning on the cryostat. Then we deposit the lung tissue onto conductive glass slides. We then use fluorescence microscopy to take images of what we have, and then we apply our matrix. And then after that, we put it into our MALDI FPICR, which is high resolution mass spec, to give us the most accurate results. We get our raw data from there and our images, and we upload that to Metaspace, which is a way for us to visualize our data, and it produces a visualization of the molecular distribution shown on the bottom left. So we use fluorescence microscopy to do a pre-MALDI analysis of our tissue. Our tissue sections are pretty big, and to make sure that we get high resolution, we want to select small areas of that tissue to be analyzed by the mass spec. So when we use fluorescence microscopy, we might see that like many airways fluorescence blue and green, as shown at the top right image, or alveoli will generally fluorescence blue, and some cells can be identified as fluorescing as red. The brightness of the fluorescence depends on the concentration of molecules on the tissue at that particular location. And we can also check the condition of the tissue to make sure it hasn't been torn and is usable for MALDI. So the matrices we use are DBH, which is a weak acid, and NEDC, which is a weak base, and methanol, dissolved in methanol. On the right, we have autofluorescence image of tissue coated with DHB and tissue coated with NEDC. It should be noted that the matrix causes a higher intensity in the autofluorescence. The solvent extracts the lipids and biomolecules from the tissue, then evaporates off, and then the matrix crystallizes with the incorporation of our biomolecules. The role of the matrix is to absorb a majority of the laser energy, and it keeps the lipids from getting degraded and it aids in ionization because the weak acid or weak base either donates a proton or extracts a proton. So our top right image shows the targeting of the mass spec, and on the right of that image is what the spectra looks like. We upload our images and our raw data to Metaspace, which is a cloud-based platform for metabolite annotation and visualization, and we get something similar to the bottom here. On the bottom left, we have just a image, a stained lung tissue after MALDI. And in the middle, we have, we can identify one compound that was found on here, on a particular area. And the Metaspace will highlight this by showing with the brighter, the more intense, or like the more concentration of that molecule will be this bright yellow, and it will range all the way down to dark blue. We can then overlap the image we get from Metaspace with the stained image, and we get something like on the bottom right. Here is a video just showing what else we can do with Metaspace. We can choose one compound to identify and choose what color it should be. And we can choose multiple compounds, and we can assign them each different colors so we can see the distribution of all of the lipids we want to see. Another technique that we're using for data visualization is uniform manifold and projection. It allows us to map clusters of ions to pixels on an image, and it produces something what looks like to the right, where each cluster is each color is a different cluster of ions. It can be used to identify lipids and biomolecules on key features such as the alveoli, airways, etc. Thank you for listening. A special thanks to all my mentors, Chris Anderton, Brittany Gorman, and Keeser Cho. And a big thanks to Pacific Northwest National Lab, the NIH, and HuBMAP. Any questions?